Good afternoon, everyone. On behalf of South African Association of Botanists, I welcome everyone to this webinar series. The name is Adeyemi Aremu. I think it's a big privilege this afternoon, and I've told myself I will try as much as possible not to take over Anina's job. I will say very little about Prof. Steve Johnson, but it's really someone I look up to. He's from UKZN, and he was the gold medalist for this current year. So without wasting much time, I will just hand over to Anina. I won't take over your job, Anina. You can kindly proceed as the chairperson. And welcome, Prof. Steve Johnson. We appreciate your time. Thank you very much.
Thank you.
Thank you very much. Good morning. I'm Anina Kutsia, and I'm here to introduce our speaker today. Prof. Steve Johnson is our gold medalist winner this year, and that's because of the fantastic botanical research that he's done through the years. He started with the work on flower evolution from his PhD and has carried that on while working at the University of KwaZulu-Natal, where he is still currently. And through the years of work, he has really achieved amazing things in this field, and particularly he has been awarded NRF A-rated scientist status since 2009 already, and is currently an A1 rated scientist, and also a Saatchi chair holder. And has received numerous awards and fellowships over the time for his research and all the work, and currently has a Google Scholar citation rate of H index of 80, which is off the charts. But all of this, I think, isn't what bothers or drives him, but it's just the process of building this field that he's so interested in, which is the pollination systems, contribution to flower evolution and speciation. And he's done some fantastic work in developing this field and making new discoveries. And one aspect of that he's made a big contribution in developing is the role of scent in pollination systems, especially in South Africa. So that's what we're going to be hearing a bit more about today, which I think is going to be super interesting, and we'll be spoiled with some great photographs as usual. So we really look forward to that. Just before we start, quickly, some household things. If the audience can just remember to switch your microphones and cameras off and keep them off during the presentation so that it doesn't distract us. And then your questions are welcome, please. So please raise your questions either at the end or in the chat. So there's a chat function there where you can just type your question and we'll address all of them at the end, where you can also then just put up your hand and ask your questions yourself. So we'll have time for that at the end. And then a final admin thing, everybody that's wants to get recognition from SACNASP for attending the webinar, please just put your name in the chat box with the word SACNASP so that we can make a note of that, keep record. Hope there isn't anything that I missed now. I think that is all. Thank you very much, Prof. Johnson. We're really looking forward to this. And you may go ahead. Thank you very much.
Okay, thanks, Anina, and hello, everybody. I chose this topic because I find it an exciting aspect of research, but also because it's been challenging. Like many other biologists, I don't have a strong background in chemistry, and yet I've come to realize that this is an aspect of research on plant-animal interactions that cannot be ignored, and so I've kind of thrown myself headlong into the field, for better or worse. So what I'm going to do is talk about some of the people that have inspired me over the years, talk by necessity about some of the equipment that we use, some of the research findings, some of the highlights and challenges. And so just before I go on, this milkweed, this Stapelia here, illustrates much of what we're talking about. It has flowers that emit sulfur volatiles that attract its fly pollinators. Its vegetative parts are defended by cardiac glycosides that are overcome and consumed by monarch butterflies, which use them for their own defense against predators. And while being eaten, the plant emits volatiles that attracts parasitic wasps that attack the herbivores feeding on its leaves. So every plant has its own set of chemicals that it uses to survive in its particular environment. The field of chemical ecology is young. This is partly because of the development of the theory, but also because of the development of the necessary equipment. The two most important founders are Tom Eisner and Jerry Meinwald at Cornell University, who did all kinds of exciting research on the chemical ecology of insects. Tom Eisner is famous for showing how bombardier beetles use hot acid to defend themselves against herbivores. And they worked together for many years. And what I particularly like is the fact that Tom Eisner had 1 or 2 pianos in his office, and they used to play flute and recorder sonatas in their office, which I find which is really my idea of a sort of stimulating research environment. Although I'm less convinced about wearing a jacket and tie to work. They were sometimes called the Tom and Jerry show. So I also put here the sort of mission statement for the International Society of Chemical Ecology, because I think it clearly explains what we're trying to do. So the mission is to promote the understanding of interactions between organisms and the environment mediated by naturally occurring chemicals. And that includes research on chemistry, biochemistry, and function of natural products, their ecological roles, their evolutionary origin, and their practical application. And I should say that this field, probably 90% of the research that goes on is practical and related to agricultural applications. And so one of the things that we've done in our lab is branch out into applied aspects of chemical ecology. Now, my own introduction to this field was a bit of a stop-start affair. I did a postdoc in Sweden. I worked at the Uppsala University field station, and scattered throughout the building was a whole lot of old and abandoned chemical ecology equipment, because Bertel Kullenberg, who'd started the field station, had by that time retired. And I sort of had the sense that this was a field, that time had passed. And it put me off chemical ecology for quite a while because of this kind of impression of this abandoned equipment. And it was actually a false impression. In fact, chemical ecology had been going on in South Africa for quite a while without me really being aware of it. So Ben Berger at Stellenbosch did excellent chemical ecology in the 70s, 80s, and 90s, even into the 2000s, mainly on chemical communication among mammals, as well as pheromone systems of some insects. Some of us in the late 90s started sampling flower scents and sending them to Roman Kaiser at Geworden. It's a perfume company in Switzerland. And he was an expert chemist that gave us scent profiles for many different plant species. So John Manning and Kim Stein and myself did this quite regularly, but many really sort of describing floral scent as part of the phenotype. So this is an early scent collecting apparatus sending material to Roman Kaiser. The big change was in 2006 when Rob Ragusa, who's now essentially taken Tom Eisner's role at Cornell University, visited my lab on a sabbatical. And Rob really inspired me to take this field more seriously. And around the same time, the lab was visited by Stefan Doetl. In 2007, I also started a SAAB chair. And Andreas Jürgens became my teaching replacement, and he was another chemical ecologist. And I was already working quite a lot with Florian Schistel, who's an orchid biologist and chemical ecologist in Zurich. And in more recent years, I've been joined by Adam Shuttleworth and Timo Pananet, colleagues who work with me on much of the research that I'm going to talk about. So with money from the Saatchi chair, I purchased the lab's first GCMS. And this is now being used to analyze tens of thousands of samples. And I was persuaded by Stefan and Andreas to go for a specialized kind of injector that allows thermal desorption of samples. So they had worked out that this device called a chromatoprobe could take these tiny little scent traps and thermally dissolve the entire sample in the injector, making the system very much more sensitive. And then in about 2012, we acquired a system for electro-antennal detection of volatiles. So this works by using a GC to separate volatiles and then to blow them over an insect antenna, and the neural signal is amplified, and you can line up the antennal response with peaks to work out what insects are actually smelling. And in recent years, we've combined that with mass spectrometry so that you can compare what an insect is smelling and immediately analyze the mass spectral signature of the volatiles. And recently, Adam and I published a paper showing that one can do this with antennal detection, flame ionization detection, and mass spectrometry all in one analysis, which has greatly improved the research capability. So going on to the actual research, the first system I want to talk about is rodent pollination. So this is really the reason why Rob Ragusa came out on his sabbatical. And ironically, it's the one system that we haven't really yet worked out. It's been a long-standing project. Many South African plants, as you would know, are pollinated by rodents. They tend to have this kind of yeasty, cheesy kind of smell. And of course, comparative biology can be very useful. So in Protea, for example, the scent of the rodent pollinated ones is quite different to Proteas that are pollinated by beetles, which have a fruity smell. And Sandy Steenhuisen in her PhD showed that beetles detect many of these fruity compounds, mostly esters and monoterpenes. And these are largely absent in the rodent pollinated species, which suggests that there's quite an important difference in the scent chemistry for rodent pollination and beetle pollination. And of course, the bird pollinated ones are hardly scented at all. So we knew that something special was going on with the scent of rodent pollinated plants. And the first system that I worked on in detail was a parasitic plant in Mpumalanga, Cypnus ficerae. Priscilla Bergoni discovered the species and alerted me to its presence. And it turned out to have a very simple scent profile consisting of two ketones, one and very unpleasantly scented one called 1-hexene-3-one, and another one found only in the nectar, 3-hexanone, which has a pleasant scent to humans. We showed that the species was pollinated by rodents and by elephant shrews. This is a video of Priscilla showing an elephant shrew feeding on the flowers. Elephant shrews, we now know, are very important pollinators of plants throughout Southern Africa, and they're actually omnivorous, not insectivorous, as was thought to be the case in the past. So we found that the rodents were not interested in the unpleasantly scented compound and were strongly attracted to 3-hexanone, the one that smelt pleasant to humans. In recent years, we've explored the general responses of rodents to a range of compounds, particularly homing in on ketones, because they're compounds that you would expect seed-feeding rodents in particular to feed on, because ketones are often products of the breakdown of fats. This graph shows the response of Namakwa rock mouse, Rhabdomus pamilio in the Cape, Rhabdomus dialectus in the summer rainfall area, and another mouse, Mastomus, which is not involved in pollination, to a range of compounds of different classes. Blue ones here are ketones, and you can see a lot of positive responses. These are confidence intervals. Any of them above 50% here indicate a positive response, and you can see lots of positive responses to ketones and to esters, but not to aromatic compounds typical of insect pollinated plants. And strangely enough, not to dimethyl disulfide, even though we find this compound in many rodent pollinated plants. Now, we have found that the response of rodents to compounds doesn't change much when you condition them by association with a sugar solution. So my sense is that these responses to these ketones, for example, at the top here, are fairly innate in these animals. And so that's how they discover these flowers in the first place. So aromatic compounds, even with training, you can't really get rodents to be attracted to them, which makes sense because a rodent following the signal of insect pollinated flowers would arrive at a lot of flowers which would not be energetically profitable for the rodents. One group of plants where we have made progress is Eucomis. Previously, Em and I showed that dimethyl disulfide was a key attractant to flies in summer rainfall Eucomis species. And then Petrovesta found that Eucomis regia in the Namaqualand was pollinated by rodents and by elephant shrews, and that they were strongly attracted by a sulfur compound methionol, which is basically the smell of fresh potatoes, the sulfur compound. So it seems that sulfur compounds may actually be important after all for rodent pollination, but not dimethyl disulfide. So we I don't really feel we've understood the chemistry of rodent pollination yet, but I think we sort of making progress. Now, the next topic I want to talk about is a long-standing interest in geographical variation and divergence among populations, and in this case involving scent. And I'm going to talk about two examples. The first is cycads, postdoctoral work of Prince Sinyu. So just before we look at that, I just want to point out what's interesting about cycads. They're brood site mutualisms, just like figs, where the pollinators basically grow up on the plant. But in this case, it's a bit unusual in that the weevils that pollinate cycads use the male cones as a brood site and emerge from the male cones and visit the female cones essentially by mistake. The females don't have nothing, the female cones have nothing to offer them, and they get lost in a maze, the back of these sporophylls, and then in that way they pollinate them. So the females essentially in them exactly copy the scent signals of the males. So Terence looked at geographical variation in the cones volatile profile of Encephalatus villosus, common species that occurs along the coastline, and found that the northern populations had scent profiles dominated by alkenes, by unsaturated hydrocarbons, and the southern populations had scent profiles dominated by monoterpenes, as well as by pyrazine, which is a nitrogen containing compound. And the northern insects showed good antennal responses to octadiene and octatriene. The octadiene we could obtain commercially. The octatriene, we had an honor student at UCT trying to synthesize it, and then they had a fire that burnt the lab down, so we lost our been able to test the octatriene with cycad insects. And the southern insects responded to the pyrazine. So first thing here, importantly, Terence did a Y-maze experiment. We had cones of the Eastern Cape plants and on one side of the Y-maze and the KZN cones on the other side, and found that the Eastern Cape insects preferred the Eastern Cape cones and the KwaZulu-Natal cones, even though they were ostensibly the same species of insects. And then using a bucket trap, number of insects caught per hour was highest for the pyrazine in the Eastern Cape, and highest for the triene, for the diene in KwaZulu-Natal, the commercially available diene. Now, we don't know if these are hardwired preferences of the insects or if this is a case of imprinting where an insect has been shown in noctuid moths that an insect may prefer volatiles of the plant in which it grew up as a larva. But it is an interesting geographical gradient that is suggestive of coevolution. Now, the next example I want to talk about is a group of insects that I'm very interested in, which are hawk moths. And we know from years of light trapping that hawk moths tend to show a bimodal distribution of tongue lengths, that they either have tongue lengths around 4 centimeters, or there's 2 species, and in fact, almost the biomass is almost entirely one species, Agrias convolvuli, with a tongue length of about 10 centimeters. So the niche, in a way, consists of either 10 centimeter moths or 4 centimeter moths. And if you look at the distribution of tube lengths of flowers in South Africa, followed by hawk moths, you can see it matches this pattern that you see with the moths, and you get exactly the same thing in Kenya. So this is an Africa-wide phenomenon, that there are 2 major hawk moth pollination niches, one short-tongued moths and one long-tongued moths. So these are some of the species pollinated by the long-tongued moths in South Africa, pollinated by Agrias convolvuli. You can see they all have similar long tubes, but they represent many different genera and many different families. The longer tubed ones, these long tubed ones with 10 centimeter flowers, are concentrated in the eastern, more subtropical parts of South Africa, and further north into Africa as well. Now I wanted to hone in on one of these species, Gladiolus longicollis, which we've been working on for many years. It's interesting because it seems to have evolved to exploit short-tongued moths in the Eastern Cape and long-tongued moths in KwaZulu-Natal and Mpumalanga. So the short-tubed ones, they are pollinated by short-tongued moths, the long-tubed ones by Agrias, and Agrias also sometimes visits the short-tubed ones, which is important for reasons I'll explain in a moment. So a picture here shows, it's a camera trap video showing a short-tubed plant of Gladiolus longicollis being fed on by long-tongued moth, but this moth is not an effective pollinator of the short-tubed one because it doesn't get into the parts of the flower. So if we just quickly go back to this previous picture, the long-tongued moths get more nectar out of the long-tubed ones than the short-tubed ones, and the long-tongued moths could visit the short-tubed ones but don't effectively pollinate them. So what would happen if you had a mixed population of the 2 forms? So across the distribution range, there's a difference in the scent chemistry of the long-tubed and short-tubed plants. The long-tubed ones have scent dominated by the monotopine linalool, and the short-tubed ones dominated by osamine, which is a monotopine, as well as methyl benzoate. And there's a zone of contact between the 2 forms of Gladiolus longicollis where you get more substantial variation. So you get these zones where the 2 forms co-occur and they keep their integrity, but they can also hybridize. So it's possible that the scent is used by the moths as a way of determining whether it is going to feed on a short-tubed or long-tubed one. So the long-tongued hawk moth, because it gets more nectar out of the long-tubed flower, should prefer that over the short-tubed ones and use scent as a way of determining that. So here you can see the difference in the scent chemistry of the short-tubed and the long-tubed plant. And in the zone of contact, you also get intermediate kind of chemistry in the introgression site where you get hybridization. So this is incomplete work, but it suggests that scent chemistry could be part of the reasons for the coexistence of these two forms. So the next topic I'm going to talk about is mimicry. It's something I've been interested in for many years. And just to point out for people out of interest that although most of us associate the field of mimicry with Henry Walter Bates in the 1850s and his discovery of mimicry among Amazonian butterflies, in fact, the first kind of real recognition of the concept of mimicry was Christian Sprengel in the late 1700s, who noticed that the South African Stapelia attracted flies, and he wrote that he thinks it smells like carrion in order to lure bottle and carrion flies. So he was essentially talking about mimicry 50 years before Bates. And this rather interesting picture is from a book called The Temple of Flora. And what's interesting is the little snake at the bottom, kind of alluding to the sinister depiction that's going on by the flower. So in animals, mimicry is almost always to do with protection, whereas in sometimes, rarely, what we call inviting mimicry. But in plants, most mimicry is inviting, so attracting a signal receiver, an animal, to the plant, and only in some cases is mimicry to do with protection. So if we look at inviting mimicry in plants, there are generally three important types of deception going on. So plants are imitating the cues that animals use to locate either their food sources or their mating partners or their brood sites, their oviposition sites. So this results in what we call cognitive misclassification. In other words, the signal receiver takes the plant to be one of these three things, and the subsequent behavior by the animal enhances either the pollination or the seed dispersal of the mimic. So the question then is, what is the role of chemistry in this form of deception? So the first example is to do with food source mimicry, but not what you might expect. Pollinators getting nectar from flowers. These are flies that feed on the hemolymph of wounded honeybees. These are millichelid and chloropid flies, and they're using wounded honeybees as a source of food. And Anne-Marie Heidak, who's been a postdoc in my lab for the last couple of years, did some very important research showing that seropegias often mimic almost precisely the chemical profile of wounded honeybees. For the GC profiles of the honeybee here, and here's the plant. You can see they're very, very similar. They elicit very similar responses in flies. So they mimic the chemical profile of a wounded honeybee in order to attract these millichelid and chloropid flies to the flowers. So this is essentially a kind of food source mimicry. And here's another remarkable discovery by Anne. This is what was previously in the genus Brachystelma, a species of Brachystelma that attracts millichelid flies, and you can see they're behaving just the same way they were on the wounded honeybee. And in this case, this mimicry is taken to another level. It's not like a seropegia flower that traps the flies and then releases them. In this case, the flies land on the flowers and they feed on an exudate, which is a sort of a mimic of hemolymph. It's a nectar-like substance that's rich in protein that the flies feed on, and in doing so, they're lured into the center of the flower and then pick up the pollinarium. So, and the four main compounds that are emitted by wounded honeybees are the same that are emitted by the flowers of this seropegia species, and these attract flies, millichelid and chloropid flies. Now, there are many flowers in South Africa that smell like rotting meat. You could call it a rotting meat syndrome. And we're not quite sure at this stage whether to refer to this as mimicry of a oviposition site or food source mimicry, because flies both feed and lay eggs on rotting meat. So it's a problematic problematic for sort of, if you wanted to have a classification of mimicry, but it's definitely a reality that many plant species, as well as many fungi, produce lots and lots of sulfides, acids, and some benzenoid compounds that are typical of rotting animal flesh, and in doing so attract flies, and in fact sometimes beetles. Now, something that is definitely mimicry of an oviposition site are flowers that mimic rotting fruit to attract Drosophilid flies. This is a nice study done by Florent Motos, who's a French postdoc who worked with me and Benny Betebier. This study was done on Reunion on a Gastrodia species of very, very tiny little flowers. And it's pollinated specifically by these Drosophilids, Scepter Drosophila. And even though other Drosophilids were common in the local community. So one of the questions is, why is the system so specialized? So Florent, and just while I talk, just notice this, that the lip of this orchid here is acting like a touch-sensitive trapdoor. So as the fly walks on the trapdoor is stimulated to start closing, and the fly will actually escape, at least this time. So the flies are attracted by 3 different esters that specifically attract Scepter Drosophila flies. And what's remarkable is this trapdoor actually resets itself. So this is a video that's sped up. So it didn't catch the fly this time. It slowly starts opening in like a Venus flytrap. And the next time the fly will, well, I'm giving it away, but it's going to get caught. Now, if you just glance to the right, this little channel in which the fly gets sort of trapped has to be exactly about 800 micrometers wide. So there are flies that are too wide to operate. Now the fly's gone in, it's getting trapped again. So the fly needs to be just of the right size to operate this pollination mechanism. So here the fly is trying to get out of the little trapdoor, and it gets out with pollinia stuck to its back. So it's a case where both chemicals and specialized morphology dictate the specialization in the pollination system. Now, what about mimicry of, or fecal mimicry of dung? Here's an example from KwaZulu-Natal of a Wembeya elatior, which has a very strong fecal odor. And we looked at the possibility that this was mimicking animal dung and attracting flies that way. It also has these markings, and we made various models, some with spots, some without. And we also looked if we could manipulate another related species that doesn't have a strong smell and that is pollinated by hoverflies. So the antennal responses of a lot of different flies were studied. This is work done by Adam. And most of them responded both to indole and scatol, these 2 nitrogenous compounds over here. But fly, which is strongly associated with dung, reacted most strongly to scatol, which is a compound known to be found in animal dung. So we put out these model flowers, and it turns out that indole is not attractive. It's scatol which is attractive, and you can see scatol alone is as attractive as a mixture of indole and scatol. And the attraction of flies was greater if the model flowers had spots. And this is because these flies tend to aggregate on flowers. So they're kind of sociable. We added indole and scatol to flowers of Wembeya krausei, which is pollinated by hoverflies. We could attract a lot of these dung flies. We almost sort of shifted the pollination system from hoverflies to dung flies. Now, I've been involved in another remarkable kind of mixture of mimicry of oviposition sites by plants, but in this case involving seeds rather than flowers. So this Restio Ceratocarium has seeds which are round and which resemble animal droppings. And together with Jeremy Midgley and some of his colleagues at UCT, we looked at the chemical ecology of this system. So this is seeds of Ceratocarium, quite different to those of other restiodes, which are not usually round and which are unscented, whereas seeds of Ceratocarium smell like animal droppings, and they also have the size and shape of animal droppings. And indeed, the seeds of Ceratocarium are carried away by dung beetles and buried underground by dung beetles. And in the sort of a 2-dimensional scent profile, the scent of the seeds is very similar to that of animal dung and very different to that of other restioes, which are mostly unscented and which emit very different kinds of compounds. So the last one I want to finish with is perhaps the most remarkable of the systems I've been talking about, because this is a study that we carried out literally using a single flower, which I think is unprecedented in the history of pollination biology and certainly in the history of the study of sexual deception. But the circumstance was dictated to us by the rarity of this plant, Isophoraceae, which was a Cape orchid, which I never expected to see in my lifetime because it had last been seen in 1966. And when I wrote the Cape Orchid book with Bill Lilford and the field guide with Benny, we really thought by that stage that the species was extinct because we had looked at it for many years and never found it. So a single plant was rediscovered by some plant enthusiasts in 2016, and it flowered again in 2018 and then disappeared altogether. And the study is on this plant and in fact on a single flower of this plant. So you can notice here that it has high fruit set. It doesn't self-pollinate. This is because it attracts pollinators. And Callan Cohen, who's an ornithologist and a very good naturalist, heard about this plant and went to observe it at a site near Hermanus. And then he got hold of me and he said he's seen something remarkable and he thinks it might be sexual deception. And this is the video he sent me, which, for reasons you will see now, immediately got me very interested. So the flowers were being visited by what looks like a wasp, but which is in fact a longhorn beetle that mimics wasps. And it's biting into these extraordinary petals over here. Its abdomen is buried, tip of its abdomen is buried in the lip of the flower. So this is from a photo from when we looked at it in 2018. These are these extraordinary petals, highly modified petals that resemble insect antennae, and longhorn beetles, when they mate, tend to bite into the antenna of the females, just like you saw in that video. And there's the tip of the abdomen nestled into the tip, into the end of the lip over there. So this is a longhorn beetle in the genus Corythaceae, and it was the first recorded males of the species, in fact. So here's a flower that I photographed when we arrived at the site, and then this is after a beetle visit. And you can see the visidia, which are attached to the pollinia, are removed, and fresh pollen maculae are deposited on the stigma. So there's no doubt that this beetle effectively pollinates the plant. But what we noticed was there was a small little deposit of something on the lip of the flower. And it took us quite a bit of time to use a needle to get this into a, this is how small it is compared to a tip of a pencil, to get it into an Eppendorf tube. And I brought it back to Maritzburg and tried for about a week to work out if this is what we suspected, whether it might be sperm of the beetle deposited by the male beetles. And eventually I figured out a staining procedure. And this is a photograph of the slide, thousands and thousands of beetle sperm. This is only the second recorded case of an insect actually ejaculating on the flower of a plant. Here's a close-up of the beetle sperm. They're about 200 micrometers, quite long, about 200 micrometers, about a fifth of a millimeter in length, the tails. So we then thought that the chemistry of the system would be interesting, and we took an extract, a solvent extract from a single flower. And we took some beetles and the solvent extract back to the lab, and Adam recorded these extraordinary antennal responses from this solvent extract. And what's really amazing is that Adam diluted this solvent extract up to 1 million times, and he could still record an electrical response of the antenna to one compound, giving this very strong response here. Problem at that stage is that we were down to the last 3 microliters of the extract, and I knew I had to get this. We were not seeing anything on our instruments. I knew I had to get this to an expert lab to identify. I took it to a conference in Switzerland, gave it to Stefan Dirtel, who took it to Austria, who gave it to someone else, who took it to the Max Planck Institute. And from this 3 microliters, Alice Fatas, who's a mass spectrometrist, using high-resolution mass spectrometry, got enough sort of information to identify it as a macrolide, which is a large cyclic lactone. And eventually this was synthesized by a colleague of his, Gerrit Weisflor. So this is a macrolide. Some of you may recognize macrolides from antibiotics. They're generally produced by fungi. A lot of human antibiotics are macrolides from fungi, but macrolides had, I think there was only one previous record of a macrolide being produced by a plant. So this in itself was a very unusual discovery. So it's a large 16 carbon ring. And this is the result from a chiral column. So this is two isomers. It was determined that it was the S form. So it's two stereoisomers. It was the S form that was produced by the plant rather than the R. So they synthesized the compound and sent it back by courier. And this is the very exciting moment where I opened the bottle of the synthesized compound and this beetle arrived. And we knew we had the right chemical formula. So the next thing that Gerrit did was he synthesized variants of the compound. So he synthesized both R and E and Z versions. So there's a Z version, the double bond. There's an E version, the 9-enolide. He synthesized the 8-enolide. And then later on, he managed to produce both enantiomers of the compound, so the S and the R, which was quite an extraordinary bit of synthesis, I think. So we called the compound produced by the plant a common name of diazolactone, and then we tested them in the field. So one of the extraordinary things about the system is the beetles are only active between 11 a.m. and 2 p.m. We put the compounds out and we successfully attracted quite a few beetles. They showed some interest in the Z8 and the E9. The strongest responses were to the racemic mixture of the Z9. And then of those enantiomers, they were most strongly attracted to the S version. So if you looked at the oxygen molecule, the one where the ethyl group is facing left rather than right. So the chirality, that is the position and the geometry of the alkene functional group are all critical elements of the function of this molecule. So here's a video just showing how these experiments were done. We made these little plastic model flowers, which we treated with these compounds. And here you can see this. Interestingly, this one was done on the Cape Peninsula, where Diazophorficaria hasn't been seen for a century, but the beetles are still there at places like around Simonstown, where the species occurred in the past. So the beetle is still there, but the plant is thought to be extinct. Now, the last extraordinary thing was in 2020, the plant had stopped flowering. It had disappeared altogether. We don't know what happened to it, but we put out our compound as a lure and we attracted 3 beetles carrying pollinia of the orchid. So even though we couldn't find any more specimens of the orchid, using this chemical method, we could determine that Diazophorficaria is not extinct and there are other plants there in the dense vegetation that just simply haven't been found, which I guess is a piece of good news. So just to finish off some of the highlights, we've managed to establish a state-of-the-art chemical ecology lab here at UKZN, thanks partly to the SAAB funding, which has made that possible, but also by extensive human expertise based on various international collaborators, as well as local collaborators, and a very nice lab group. And we've made many scientific breakthroughs as a result. There are certainly challenges. One I haven't mentioned there is students obviously have a fear of chemistry, which is makes it sort of intimidating for some people to get involved in this field. The equipment has a certain lifespan. Eventually, it's hard to get spare parts. At the moment, because of the Russian invasion of Ukraine, there's a terrible shortage of instrument-grade helium that's affecting us. The problem of reliable power supply. Mass spectrometers don't like being switched on and off, and so breaks in power supply are fatal to these instruments. And then the other final challenge is specialized chemical expertise, particularly ability to do chemical synthesis, is in short supply, not only in South Africa, but in fact, worldwide. So yeah, so finally, just to thank all the lab members and my other research collaborators for their partnership with this research. So thank you.
Fantastic. Thank you very much. That was excellent. So interesting. Right. So if there are any questions, please feel free to put them in the chat box, or just raise your hand, or just unmute yourself if you want to ask any questions. I'm just going to check here if there's any over here in the chat box that I missed. Seems like the audience really enjoyed the talk.
Should I keep this PowerPoint up just in case?
Yeah, sure.
To refer to a slide, yeah.
Yes, that's fine. Great. Glynis, yes, please go ahead.
Thanks, Steve. That was really fantastic. Gosh, what an amazing array of different kinds of plants and pollinators and scents. Fabulous. Thank you. I wanted to ask you, so I know you're interested in the evolution of these interactions. What is your sense of how something like that would evolve? Do you think it would be over a long period of time? Would the plant very slowly accumulate those kinds of chemicals and be rewarded for it? Simply a natural selection sort of effect? What's your sort of thoughts on that?
Yeah, that's a very good question. We think that in some cases, I mean, in eucomas, for example, we can detect some of the compounds in trace amounts in the other members of the lineage. So for example, the rodent one has trace amounts of the dimethyl disulfide that attracts flies. And in fact, the fly one has trace amounts of the methionol that attracts rodents. So probably it's a case of upregulation in some environments in which those animals are available as pollinators, giving the plant higher fitness. So in most cases, I think that the sort of molecular machinery is already in place, and it's a kind of a case of sort of upregulation rather than a mutation for an entirely new sort of group of compounds. Although with the disulfuric, for example, I think that macrolide is a novel mutation. I don't think, well, I can't be sure, but I don't think it occurs in the rest of the lineage. We had a very interesting case in Satyrium where we found a form of Satyrium longicorda, which is a common moth. In fact, it's the one in the photo. So Satyrium longicorda is pollinated by moths, and we've found a population that is pollinated by oil bees. And production of oil leaves a chemical signature compound called diacetyl. And in fact, when we looked at Satyrium longicorda, we found trace amounts of diacetyl. So it's possible, for example, in Satyrium that the two spurs represent an old ancient sort of adaptation for accommodating the two front legs of oil bees. And that was kind of, that oil system was lost, but the kind of genetic machinery or the genetic architecture remained in place. And then under certain circumstances, it was switched on again or upregulated. And you had the shift back to oil bees in Satyrium. So yeah, I guess the answer is in most cases we don't know, but we've got some clues from these kind of comparative biology.
No, it's fascinating. Thank you. Sure.
Thank you very much. I see there's one question about the recording. This presentation recording will be available from the from SAAB's website. I'm going to ask Bob to ask his question, and then I'll address the others in the chat box. Bob, please go ahead.
Thank you, Steve. That was fascinating to a complete layman like me. The last example with the longhorn beetle and the thought to be extinct orchid, but no longer extinct because the longhorn beetle was found with its pollen. Has that line of evidence ever been seen before to prove that something isn't extinct that was thought to be extinct? It's a remarkable and very beautiful demonstration.
I don't think so. I don't think so. I think it's probably the first. Well, I think that people have used chemicals to establish that pollinators are still in an area. So, for example, when they do work for mine reclamation in Western Australia, where there are lots of very rare orchids that are sexually deceptive, they'll put out the compound that attracts the specific wasp, and then they will use that to establish that the wasp occurs naturally in that area. But I think that using a chemical lure to attract an insect and then sort of barcoding the pollen to establish that the plant is there, even though it hasn't been seen, I suspect is the first case.
Remarkable.
Yeah.
Thanks.
Thank you. Coral, I see your hand up. I just want to address these questions in the chat quickly. Marion Mayer is asking out of interest, I think he's asking you, Steve, whether you have only first-year university chemistry.
Yes. Yes, like most biologists, I would say.
Yes.
Yeah.
We should maybe consider giving our first year or our undergrads a bit more chemistry so that they don't have that fear.
You know, just to comment on that point, I mean, I think like all things, like statistics, chemistry, when you don't have a reason to use it and you study it, you don't really realize it. But then when you suddenly need it, then you dig deep and you learn.
Yes.
So I think the problem is for first years, they learn chemistry, but they don't see the application.
Right.
So that's why you have to go back to the drawing board, I think.
Coral, please go ahead and ask your question.
Hi, Steve, can you hear me?
Yes, hi.
Hi, Steve. Steve, just a general comment that came to my mind during your talk. So the mission of this chemical ecology is to study the ecology of interactions and how they're mediated, I guess, by chemistry. But I'm wondering, you know, ecology really means species interactions. And I'm looking at these photographs and I'm thinking, well, how are these bouquets of chemicals influenced by the presence and absence of other species?
Mm-hmm.
I'm thinking also in terms, because I've come to realize that fungi are everywhere and they also produce chemicals. And I'm just curious to know what the future of this type of research for you is, because you've studied so far pretty specialized interactions with pollinators, looking for that key molecule that provokes the pollinator. But I'm just wondering if these are also then influenced by the presence and absence of other species, and whether there's multiple species. And
you mean whether they're sort of like induced by herbivores and that sort of thing?
Yeah.
Is that what you mean?
Or in my case, fungi. That's what I'm thinking about. But yeah.
Yeah.
Does that make sense?
I think that is something. It's well known for people who study herbivory that the process of herbivory completely changes the chemical profile of leaves, and very often the release of those kind of, those C6 leaf volatiles is often used as a cue for wasps to attack herbivores and so on. We've wondered whether something similar might happen with flowers when they're damaged, but it's a little bit less clear what is happening. But yeah, I mean, if you're talking about whether the presence of a particular fungal associate might affect the floral scent, think that nobody knows. Yeah.
Okay.
Yeah.
Well, the other aspect could also be a pollinator influencing, because pollinators also leave chemicals on the flowers.
Yeah.
As a form of signaling.
Yes. Yes. Good. Yes. I forgot about, yeah, they do. The bees leave kind of fingerprints.
Yeah.
Chemical fingerprints that other bees use to realize that there's no point feeding on that flower because another bee has just drunk all the nectar and so on. So yes, I think that is the case. I mean, it gets very interesting when you're dealing with fruit volatiles that attract drosophilids.
Yeah.
I mean, that's a very complicated thing where the yeasts are, it's actually the yeasts that are producing it's not the fruit, it's the yeasts.
Yeah.
And in fact, the yeasts
it was actually the example that came into my mind. Yeah.
The yeasts need to be dispersed to other fruits. So in fact, the yeasts are doing the signaling for their own.
I was thinking that in terms of fungi. Yeah.
Yeah, it's quite remarkable. Yeah.
Okay, that's my point. Okay, thank you.
Thank you. Eugene is also asking whether you can do similar things with browsing mammals and plants potentially messaging others. I assume he means using similar studying techniques that you've done.
Yeah. So I am involved in large mammal responses to fruits at the moment. But Adam, working with some large mammal biologists, has done some quite interesting work with rhinos and also with elephants, showing that leaf selection by large mammals is often based on the smell of the leaves. So they decide what to bite based on smell, which makes sense when you think of the fact that a lot of these antelope feed at night. They probably doing their leaf selection by smell. And in fact, a lot of the camera trap footage we get of antelope shows that before every bite, the leaf is kind of sniffed. So I think that volatiles are absolutely critical for herbivores to determine palatability of leaves. And as I said, I'm currently looking at acacia fruits and the role of volatiles in attracting large mammals. And that's a rich field of research with lots of potential. Yeah.
Thanks. Just one question from Ethan Newman quickly, and then I'll move over to Nathan. Ethan asked, have you figured out how to measure the phenotypic selection on scent? Would be particularly interesting with reference to the hybrid zone of Gladiolus longicollis ecotypes.
Yeah, that's a good question as well. I think so. Phenotypic selection would mean measuring the volatile profile of each flower and correlating that with some fitness measure like number of seeds produced. I think it's feasible in the Gladiolus system. I think the Gladiolus system we should continue to explore. I think it's a fantastic system to look at that kind of work. The rate limiting step is, the time taken to analyze each sample. You're dealing with 40 minutes on a machine and probably an hour of work to do the scent profile for a single flower. And to detect phenotypic selection, you typically need 50 or more samples. So yeah, it would be fantastic to do it, but it's quite difficult and quite time intensive. But the hybrid zone would be the perfect place to do it. Yeah.
Right. Thank you. Nathan, please go ahead with your question.
Thank you very much. And thank you to the speaker. It was an absolutely fascinating talk. And I, as just a simple chemist, felt very proud that you could have achieved what you did achieve in the biological sense. So thank you for that. It was wonderful. When I look at these compounds in general, I see terpenes and other volatile substances, and then I started wondering to myself that if it were a mammal or a human, the reason why these things are volatile is that it gives you an ability to smell via your olfactory epithelium what that smell is. And I started wondering to myself, how does the insect perceive that volatile in terms of which I just assume would be a sense of smell? in order to do that. Can you possibly enlighten us as to how that mechanism takes place?
Yeah, I'm not a neurobiologist, so take what I say with a pinch of salt. But you probably know in the human genome, the sort of olfactory genes, the genes that code for the proteins that interact with scent molecules for scent, is the largest family of genes in most humans and most mammals, in fact. So the scent of vertebrates is achieved by having this large sort of group of olfactory proteins, and it's very similar in insects. Except that the sort of mechanism is slightly different. So in small mammals, for example, there's something called the vomeronasal organ, which basically sort of is for smell of polar in oxygenated compounds that sort of occurs at the back of the nose. In the case of insects, there's the antenna usually has kind of pits, so areas, sort of little holes in the antenna, and the scent sort of diffuses into that chamber and interacts with these proteins that then result in the firing of a neuron if there's a kind of a spatial fit between the protein and the volatile. But it's not as simple as that. So there's further processing that happens on the brain side. So for example, sometimes a particular protein may react with more than one compound, and it might be the kind of signal from two different types of sensory systems that results in the perception of scent by the insect. So yeah, it's basically all about taking in the volatile through these pits, the volatile interacting with the protein, and that causing the firing of a neuron and a signal to the brain where it's further processed. But that is way beyond my
Thank you very much. It's fascinating. Again, proving chemistry.
Fantastic. Thank you very much. I think it's about time for us to wrap up, but this has been really, really interesting, and we enjoyed it, and we really appreciate your time in preparing this presentation. So thank you very much for that. Thank you also to the audience for the attendance and the engagement. That also makes it really fun. Thank you very much. So this recording will be available on the SAAB website. So yes, I think we're going to finish off now. I don't know if Adeyemi, you wanted to add anything at the end? Great. Otherwise, I'm just going to finish off now. Thank you very much again, Steve, and thank you for everybody that attended.
Thank you.
Thanks.